You want a worksheet out, we're going to be working the back side of worksheet 13A. Kind of, we went through the process of transcription. That's where we copy DNA to mRNA. 13.2 is going to take us through translation, which is going from mRNA to DNA. They both take place at different places in the cell. The transcription that we looked at last time happens in the nucleus. Remember, the DNA is not allowed out of the nucleus. It's protected in there. It's sequestered in there. But it's also trapped in there. And we use the message from DNA in cytoplasm. So we need to make a working copy, so we transcribe that into messenger RNA. We take that out into cytoplasm, where it binds up with ribosomal RNA, the ribosomes, and then the two together set a pattern and read the pattern to stitch together polypeptides. Remember that we read the genetic code 3 letters at a time. Okay. Anybody else need a worksheet? But the cell actually reads its messenger RNA. It reads it in 3-letter increments called codons. Codons code for amino acids. So it sounds like what it does. There. Anybody else need one? Give this a ring, Kirsten. We have that table to fill out. We'll do that just a second here. But we read them 3 letters at a time, so you'll hear people read them and you'll see them written in 3-letter blocks. A-U-G, C-C-A, A-T-A. They're phrased like that because that's how the cell reads them. Okay. So first thing we need to do, we make DNA into messenger RNA, we transcribe it, and then we will turn it into, we'll use that transcribed message to stitch together a chain of amino acids, polypeptides. Amino acids are otherwise called peptides, so that's another name for it. So when you string together amino acids, you're making a polypeptide. And with a little folding, that polypeptide can be a protein. Most proteins in organisms are enzymes, and their function is determined by their shape. And their shape is determined by the order of the amino acids. So as they're formed, they start to fold up. Okay. And as they fold up, then they become proteins. The building blocks look like this structure on the left here. And let's see if I can get... Yeah. So these are the building blocks. This is a generic shape. All amino acids have this green part in common, they have this blue part in common, and they've got a hydrogen here. What's different between all of them is this right here, and that's the R group. The R group is never an R. It's always something else. So we have 20 different kinds of amino acids. They're all the same except for this part, which is called the R group in the generic form. So alanine has CH3 for the R group, and serine has CH2OH for the R group. When we assemble amino acids, the ribosomes bond them together. Because they're peptides, that's the other name for them, and the bonds that hold together are called peptide bonds. Okay. Make it work. Okay, I said that stuff already. Okay, so here's what it looks like when we lay out messenger RNA. Let's see, we've got AUG, AAC, UCU. That's how they read them. We always read messenger RNA, so you're always going to see U's in here instead of T's. And it'll always start with AUG. That's just kind of a given. AUG is a start codon. It's what the ribosome looks for when it attaches to messenger RNA. Okay? This is the easiest way we've learned, or the easiest way that I've found anyway, to read amino acids. We have some kind of table that takes those three-letter codes and turns them into the name of the amino acid. And there are different forms. Sometimes we see this as a table, and it'll say on the left edge, first base, top says second base, the right says third base, and you read them, you know, AUG, A is the first base, U is the second base, G is the third base. This one you start in the middle and work your way out. Okay, so the first one is here in this ring. So if I'm doing AUG, I pick this one. Second base is in the medium-sized letters. Just work my way out. A, U, and then G is right here. Okay, and I know you can't see this on this size print, but that says methionine. Okay, and methionine also is a start codon. So if it's at the beginning, it puts methionine there, and it starts there. If I see AUG in the middle of a strand, it just puts methionine in. Okay. Now, it turns out if we go through and try to figure out how many possible combinations there are of A, C, U, and G, if I've got three letters, I just do 4 to the third power, which is this, 4 times 4 times 4, so 64 possibilities. We already said there's only 20 amino acids. So some of those amino acids are coded for by more than one codon. And we can see that especially places like this, where GUU codes for valine. GUC codes for valine. GUA codes for valine. GUG codes for valine. Any one of those will do that. Okay? Some are only one. There's your possibility there. And some don't code for amino acids at all. Like I go up here and here, UGA and UAA and UAG all code for stop, which is an instruction. It's not an amino acid. When a ribosome runs into a codon that says stop, it lets go. It releases the mRNA and it releases the polypeptide strand. So sometimes they're instructions. There it is, just a little bigger. This wheel will be provided for you in multiple ways on the quiz and on the test. On the gizmo, if you worked ahead, you saw this was on the back. So some of you already figured out how to use it, and if you didn't, no biggie. Okay. So what do ribosomes do? Ribosomes basically read messenger RNA. And as they read it, amino acids are brought in the correct order by the transfer RNAs, and the ribosomes will make sure that the correct amino acid and transfer RNA are in the right spot. And if they are, it will remove the amino acid from the transfer RNA, send it off on its way to go get another amino acid, and it'll attach that amino acid to a growing chain of amino acids. So it basically reads mRNA. Again, the order of the amino acid codons on the mRNA determines the order of the amino acids. The order of the amino acids determines how the protein folds, and how the protein folds determines its function. This process of going from messenger RNA to protein is called translation. It's a big change. And here is, we saw a video of this the other day where we made messenger RNA and it spooled out into the cytoplasm and then it was read. So this is the book's version of this. It's not entirely accurate because before it would come out, we have to put a telomere on the end of this. So a telomere might be a bunch of repeated bases. Like a whole big string of them on the front and the back. The nucleus looks for that telomere before it allows it out of the nucleus. If that telomere is not on there, on either end, it won't be allowed out, because that's one of the signals that the mRNA has been processed, that we took the intervening stuff out of the RNA, because the DNA strand that we decode has good stuff in it, and then it's got a bunch of stuff that we don't need. So we have what are called express segments or exons, and then we have intervening segments or introns, and we want the introns out. We don't want to decode those if they don't mean anything. So that's got to be processed, and then it puts a telomere in each of them. The telomere is a signal to the nucleus that that processing's been done. And then we send it out to the cytoplasm, and the ribosome is going to look for that telomere, that series of letters. If it's a bunch of A's or a bunch of T's or whatever it is, it's going to look for those repeated letters before it reads it, because otherwise it won't know that this message is complete. All right. And this is what it looks like on the ribosome level when it's bounded up to a messenger RNA. So we got the messenger RNA here. We got the ribosome here. It's two different units, so we've got the blue at the top and kind of the tan color underneath here just to distinguish between the two. And then we've got different transfer RNAs coming in. The transfer RNAs have to match up to a complementary codon. So this AAA, for example, is a codon. This UUU will match up to it. Okay, this is called codon. This is the matching opposite, so this is an anti-codon. And that's kind of how we make sure that the amino acids are in the correct place. Each kind of amino acid, or each kind of transfer RNA carries only one kind of amino acid. This kind only carries lysine. It only bonds up at the sequence that matches its anticodon. So AAA only pairs up with an anticodon UUU, and the tRNA that has UUU only carries lysine. So by that mechanism, AAA will only code for lysine. That's how that works. As those are brought in and they're matched up, the ribosome will release them as they come in here. So this will be released, and this will be bonded to this one. And then this will move down along the messenger RNA. Okay, and so this whole blue section will move over, and then it will be AAG, and then UUU, and then it'll remove this tRNA, let that one go, and the UUU will be the next one, and then it'll just stitch all these together, and they'll kind of feed out the top of the ribosome. So far, so good? We actually haven't answered anything yet, I don't think. Because this is kind of a skill one when I get to the back page. All right. And it'll continue to do that. And you can see the polypeptide is growing here until it reaches a stop codon. And at the time it's a stop codon, it'll release the growing polypeptide strand, and it'll release the messenger RNA. And then it might go back and reattach to it and read it again. Okay, so this whole process of going from DNA to messenger RNA, and from messenger RNA to protein, goes back to Watson and Crick. And we've modified it a little bit then, but we still kind of follow that kind of general idea. So we turn DNA into RNA. We copy RNA from DNA. Remember what that process is called? When we copy DNA to RNA. Transcription. And when we use that RNA to build a protein, what do we call that? Another vocab word. Translation, yeah. You guys feel a little self-conscious about that microphone? You're like really quiet.
Oh, I didn't even know it was coming.
Actually, for most of you, it won't pick you up. The ones around, it will probably pick up the front row.
Okay.
So the rest of you are safe. Okay, so this is the central dogma. Dogma is kind of like this, kind of, everybody accepts this. It's something that's so universal we don't even question it. We probably should, but we don't. And that is, we take DNA, we use DNA to make RNA, we use the RNA to make protein. And if I were you, hint, hint, I would know that. That would be helpful sometime later on some kind of assessments. Know that we go from DNA to RNA to protein, and to know that transcription and translation, how they're involved in that. Okay? Should we switch over to the worksheet? All right. So I'm going to, did I do that too quick? I'm going to back this up. I want to get back to that wheel. And then we'll go to worksheet, back to the worksheet. All right. So it says use the mRNA codon table to complete the following. Or you can use page 367 in the book. Or you can search it on the internet. You might find a different kind of wheel or table on the internet if you go looking. Okay. And so the other thing we should probably do is just review the complement thing. So does everybody know how to copy DNA to DNA? A goes with T and C goes with
G.
G. Okay. So can we just do, I'll try and write this a little cleaner. We'll do DNA to RNA. Okay. And I'll put them in the same order as the worksheet has them. A, T, C, and G. What goes with A?
U.
U. Okay, remember that RNA doesn't have T's, it's got U's instead. So it's like a placeholder. So if you can't remember that, just do it like DNA, then change the T's to U. T goes with A, C goes with G, and G goes with C. Now something else that would be helpful that's not on your worksheet, and you can fill this in on the margin if you want, is to go from RNA to RNA. Because you're going to be asked in the questions about anticodons. And RNA to RNA would be the difference between matching a codon to an anticodon. So U goes with A goes with U, G goes with C, and C goes with G. Okay, so that's for the table on the upper right of the backside worksheet A. It says find the complements. So you can add that one in the margin to the right there. Okay. So question 10. What are these words? What do they mean? The parts of a protein, and what are the individual names called?
Bases.
Nope.
Codoners.
What's a building block of protein?
Phosphate.
2 words. Starts with AA.
Amino acids.
There we go. So these words, these are all amino acids with 2 exceptions. So asparagine, lysine, serine, arginine, valine, those are all amino acids. The exceptions, right here and right there. Stop is not amino acid. Stop is an instruction. That's not. No, it's a codon, but it's an instruction rather than an amino acid. And there's one that does double duty, and that's this one here. Methionine codes for, methionine is coded for by AUG, and it's also a start codon. So if it's at the beginning sequence of a strand of messenger RNA, it's where the ribosome attaches. If it's in the middle, it'll just read methionine. At the beginning, it's also the start. Okay, so let's see if we can do this. A shortcut for you, by the way, and something biologists often do, because we're lazy, is that we will use the first three letters as abbreviations for these amino acids. Okay, so methionine becomes MET. Arginine becomes ARG. Valine becomes VAL. Okay, there is a couple we need to be careful of. Okay, so asparagine and aspartic acid both start with ASP. So you might want to do ASP and then capital A, so that we know which one it is. Okay. And so that'll kind of speed things up as you're writing them. You don't have to write the whole thing. And sometimes it's hard to read. You can get the first letters if you look, but it's hard to read what the word is, and so that's helpful. Stop, just write stop. But the other ones you can use the first three letters. Okay. What's AAA code for? Where do you start reading? What was that? Lysine?
Yeah.
Okay, so first letter, second letter, third letter, whoop, is right there. And that's lysine. You can do LYS if you want. Okay?
Wait, so it would just be A or B, G.
What was that?
Why would it be?
So,
Oh, never mind.
okay, we're on number 11,
Yeah.
and we're reading AAA. And so we always start in the middle, and we work our way out to the words. Okay? So let's see if we know how to do this for sure. Whoop. Maker's board. I went the wrong way. One more. Okay. Decode GCC. GCC. Gary Charles Charles.
Alan.
GCC. So I work my way out from the middle. G, C, C, and then it's alanine, and you could put ALA. Okay? I'll start in the middle. Okay, number 12, work backwards. So what's the codon for tryptophan? Give that a try before you yell out. Don't just yell it out. I want people to try it. Can you do this? So look for tryptophan and see if you can get the code. Give you a minute to find it. Tryptophan. Anybody need help finding tryptophan? Everybody find it on the wheel? Raise your hand up if you see tryptophan. Okay, I want to know that you can see it.
Another food company, Australia, all the white girls like. No,
I know not of what you speak.
It's a white girl brand.
Oh, is it?
Type of boot that they all love to wear in the winter time.
Oh, that's what you're hinting here?
Yeah.
Okay.
Oh. We're talking about.
No, I'm talking about tryptophan, not anything else. Jeez.
Everybody's trying to figure out what you're talking about with tryptophan.
These cornies.
I didn't know they were Australian.
Ever.
Okay, how many people think you got the code?
It's
How many people think you got this? How many people are really lost?
use
Okay.
instant.
Really, really lost.
They could turn like dad jokes.
How is that a dad joke? It's not even a dad joke.
Because it was
You've
Dad
been,
joke's a pun.
my dad.
you've been in here all year, you know that's true.
Yeah.
Yeah, no, but a dad joke's gonna play on. It's like this is like the riddle.
Okay, so we found tryptophan. Okay, so we read it from the middle out. First letter is U, second letter is U, third letter is, second letter is G, I'm sorry. First letter is U, second letter is G, third letter is G. Read it from the beginning out. Yeah.
So what would you do if it was like cysteine? You know that one?
This one here?
Yeah, so what would you do if it's like
You got multiple correct answers there. And
Oh, that's
that turns out to be a good thing to ask because one of our upcoming problems asks for multiple.
Yeah.
So for alanine, what are the codons for alanine?
Wait, so do you have to both? Or
You have to put all of them unless it doesn't tell you that. So in number 12, no, not 12, number 13, what are the codons for alanine? That's a plural. So you just list out letter combinations for alanine. Okay. Give them a try. We're here. It's safe now. You're not getting marked wrong if it's wrong. Yeah.
GC, G, G. GC.
Well, hang on, let people do it first, and then we'll come back to you. Hang on. So the first one she said was right, that's a hint. Try them, guys, if you're not sure what to do, try them, and then correct them. So write them in faintly if you're not sure what to do. There's gonna be four separate three-letter combinations. So we're answering number 13 right now. If you're confident, just go ahead and write it in. If you're not confident, write it lightly. So here to give us, which one did you give us? GC. So now give me another one. Yeah. GC is another one. Give me another one. GCA. There's one left. You got it.
The GCG. Oh,
GCG. GCG is left. Okay. All right. So are we good through number 13?
yes.
You have everything answered through 13. Okay. Do 15 first, then 14. This next couple questions is just to show you my stability has been in the works for more than 10 years. Well, I put the problems in backwards, 14 and 15, way back when. Realized the first year they're backwards, I need to fix that. And I've never fixed it. And every year I do the same thing. Do 15 first, then 14, because I put them in backwards.
Are you ever going?
Well, judging by the last 10 years, probably not. It's probably been more than 10. I think it's 10 years, it's probably been 15.
Wait,
The worksheet isn't exactly the same as it was, so at some point I copied and pasted it in here.
did you like forget about it or?
I forget about it, yeah. So do 15. No, because I'm not in there right now. This is why I forget. What's the codon for leucine? Okay, here's the problem. Right? That you were concerned about before, wasn't it?
Oh, I get it. Okay.
Yeah. So it says, what's the codon for leucine? What's the issue?
Oh, there's two set.
If you are not told to do multiples, in this case it says, what is the codon? No S. There are several correct answers. Pick one of the correct ones.
You.
Ooh. Is that accusatory or is that a guess?
That's a guess. Yeah.
Okay. And once you got a codon, then you should be able to find the anti-codon, which should be complementary. And of course, you already went in and did the RNA to RNA, so you can find the complementary anti-codon. Okay, now we do it at a time.
Oh, yeah, how
Kind of running a little long.
do you find the answer?
How do you find it's complementary to the codon? So, for example, AUG is the start. That's a codon.
Yeah.
The anti-codon is going to be the complement of AUG. A goes with
Oh.
U goes with
Okay.
A, and G goes with C. So the anti-codon for AUG would be UAC. You're just finding the complement. And if you did the RNA to RNA in the little box in the upper right, you're all good to go. So just copy back in the complements.
Wait, so
And any of them, any of the codons are acceptable answers for 15, and so that means any of the complements for those are acceptable for 14. Yes?
wait, so in the diagram, like, which ones are the codons and which ones are the anti-codons?
In the diagram there to the right,
Yeah.
messenger RNA is always codons, transfer RNA is anticodons. Okay. What the cell reads is messenger RNA, so that's where the codes are. The code is in the messenger RNA, so that's where the codons are. All right. Are we okay, 14, 15? 15, 14?
Yeah.
Anybody feeling like they need a hint? We're getting kind of down there in time, but I want to kind of switch over. Okay, transcription, translation, I'm going to leave those definitions for you. Remember they're vocab words, so you can get the definitions out of the vocab. So if you don't have them, leave them out while we do vocab. Okay? Number 16, list amino acids in order by the segment of mRNA. Just draw a line through every three. So AUG, put a vertical line, UUC, another vertical line, AAA, vertical line, CUG. And then just read them in order. What's AUG stand for? What's UUC stand for? What's AAA stand for? What's CUG stand for? We're going to be able to do that?
Hey, what? Say that again, a little slower.
Okay, so the mRNA strand. Every three bases separate them out with a line.
Okay.
So you have it like AUG, with a line. Oh, that was psycho. I got a little close to the bottom of the board there, I guess. AUG, UUC, AAA, CUG. That's what I mean by putting vertical lines in. Then you can read them three at a time. And then just list amino acids. All right, 19. We did this already. Might not have written it down. The central dogma of biology states that information is passed from, where do we start? DNA. And we copied the DNA into a message called RNA. And then when you use the RNA then to build a protein, that's the central dogma.
Whoa.
Okay? And then what I might do is take transcription and draw an arrow down to 2, the first 2, TO, and translation to the second TO, so you know that that's process of transcription. Number 20, I'm just going to give you because we're short of time, and it's not really on the quiz or the test anymore.
Yes.
An example of an exception to the central dogma of biology is a retrovirus. Retrovirus. Retro, like going retro.
Oh,
Okay? It's not an old virus. Retro doesn't mean old. Retro means go backwards. So retrovirus goes backwards. It goes in you as RNA. The RNA makes an enzyme that transfers the RNA back to DNA, which is kind of weird. Okay? It goes from RNA to DNA, and then it hides in your DNA for a while. And then later, something triggers it, and it goes from our DNA to RNA to protein. So at first, it goes backwards. So retrovirus. R-E-T-R-O-V-I-R-U-S, retrovirus. Retro means to go backward. So the word retro, you think it means to be like the 90s. It's a contraction of retrospective, which means to look backwards. So it's an example of English cheat.
wait, so it's is a something just with like something, which is
Okay, so an example of the exception to the central dogma of molecular biology is a retrovirus, like HIV.
oh, geez.
So you've heard that people sometimes are exposed to HIV, but they don't get sick for a while. And then sometime later, maybe months later, then they start to get symptoms of AIDS. So the AIDS doesn't happen right away. The HIV goes dormant for a while. And that's what's happened is it's gone into your body. The RNA has transferred its RNA. So basically, it makes an enzyme that codes its RNA back to DNA. And then the DNA hides in your DNA for a while. And then something triggers it later to become active. Then it goes from DNA to RNA. Okay, so an example is a retrovirus like HIV, which copies from RNA to DNA. Yes.
So like, yeah, so people contract HIV from people, but how do those other people get to begin with?
No. So it's always passed from person to person. Think originally that it was from the consumption of monkey blood. So there's always this scatological stuff where people make jokes about having sex with monkeys because it's a monkey virus, and it's a mild monkey virus. It's like a flu in the monkeys. But that's where it started. People eat monkeys and ritualistically sometimes drink monkey blood, which has not been cooked, and it basically is exposure to blood that transmits it. Yes. Yes. Yes.
So you read
the AUG on the chart then? Or-
Yeah, you read it on the chart, and the chart, if you don't have the copy there, it's on your gizmo. And just as a check,
That's
the
why.
first one is methionine, or you could write MET. Yes,
What's 19?
what's 19? Information is passed from DNA to RNA to protein. Any others?
Wait, what did you say 16 was?
So 16, you're going to have 4 answers because there's 4 amino acids. Just as a self-check, the first one is methionine. If you're doing it right, you get methionine for the first one.
Okay.
But we don't read it off the messenger.
You read it off messenger RNA. Yeah. So this wheel uses messenger RNA. This is messenger. When it's in a straight strand, that's mRNA, and that's what you read on here. So when you if you try and read DNA, it blows up right away. That's a sign you're doing the wrong thing. So you try to read DNA, there's no T's. You don't want to do what some people do, and they just switch all the T's to U's and read it anyway. That'd be the wrong thing to do.
So how many vocab things around?
10. Oh, the unit exam, they're kind of in problems. There's not a vocab section.
So during the quiz next time.
The quiz next time, there'll be 10 vocabs. There's 5 from 12 and 5 from 13. So it's actually the 5 vocabs that we had, minus the scientists, that's 5. So then we added 5 of 16. Okay, so I want to make sure we can do one more skill here before we switch. And so let me get a whiteboard up here. I was going to do this. I don't think the recorder will go back and record the smart report, so I'm going to have to come back here and do this. Okay, so what we would do, change the light down here. Don't like that one. Oh, there we go. All right, so the skill then is we'll give you a section of DNA. Okay. And ask you then, so we'll give you this and ask you to find the complementary mRNA, and then use that then to do the amino acids.
And then that's where the wheel comes in.
And that's where the wheel comes in. Okay, so go ahead and do that. Okay, do it on some blank scratch of paper you got in the margins of your worksheet, or you can do it on your gizmo, or you can do it on anything else. I just want to make sure you can do it. The wheel you need is on the back of the gizmo. Right there. Okay. Take a couple minutes, make sure you know how to do this. You are going to be asked to do this on the quiz. You're going to be asked to do this on the exam.
Exam on Thursday.
The exam is the next class period I see you.
The whole unit exam.
No, the quiz, I'm sorry, the quiz is next time I see you.
Oh,
And then 2 times after that is the exam. But we will start the next chapter in between.
wait, and
We're kind of, the snow days are making us run, we're way behind because of snow days.
will that
That's not your fault, that's not my fault, it's just what it is.
so will that chapter in between be on the
It will not.
oh,
The unit exam will have 10, 12, and 13. Try this. AUG, CC, oh, yeah. They're not supposed to be used there.
Yeah, it looks like
Let me fix that. That didn't fix it. It's wrong color.
oh, that would be
That would be a problem.
oh, yeah.
All right, now you can do it. Remember, you gotta turn it into mRNA before you read it. So
No, T is A.
Oh, it's right over there.
yeah, it's in that order. So you don't have to do another step. Okay. Guys, so we're clear?
Yeah.
We're gonna go DNA to RNA to proteins. Bang, bang, bang. You don't have to do extra steps in there. So once you switch DNA to RNA, go ahead and read it on the wheel. Okay, we're trying to, I know there are common things that people do wrong. I want to make sure that we kind of get rid of that right now. Anybody feel like they want to go up and put the RNAs up?
I can do it.
You can do it. There you are.
No. Yeah.
All right, check his work. You do it right?
It's a little squeaky back there.
Okay, if somebody's got amino acid they want to put up there, you can come up and do that.
Oh. Hold on. That's nothing.
All you gotta do is one. And you can put just the first 3 letters.
Let's see the. No, the last letter, because it's words.
We just need the first 3.
I need to see the.
That's right. It's sometimes helpful if you just do the first 3 letters, because you can make those out. Sometimes you can't make the rest of the word out.
So you got the T and then I think it was.
It is. So that's METHONINE. Do we want to do the next one? How about I do it? I don't want to do it. What do you think?
Oh,
Well, I want.
Gracie.
Oh. We only have to do the first 3, right?
First 3, yeah.
Ooh, this is fancy.
We want to do AUC. Okay, okay, spread it out. You want to do the next one? Okay, got it. We'll figure it out if you weren't. All right. They all check out?
yeah.
Okay. So let me ask you this. We have 20 minutes left.
Refresh.
How many of you are done with the gizmo? Do you think it would be more beneficial to run through the vocab or have more time for working on the gizmo? Are we okay working on the gizmo?
Okay, well,
If we had a half hour left, I'd do the gizmo first, but we have 20 minutes. So go ahead and work on the gizmo.
Wait, what?
If you don't know the vocab yet, you know where to practice, right?
Wait, so the vocab on the test, which 5?
So if you go into Canvas,
Yeah.
there are links to the 12, 13 points. Both in Quizlet and Kahoot. So if you think you need to practice more, then do the gizmo more. Go ahead and do that. There are 12, 13 practice sets on both vocab places.
Oh, this is a really good picture.